Assalamualaikum viewers, welcome to my channel Genome Study. If you are new on my channel, please subscribe and press the bell icon so you will get the notification of all the new videos. When we clone the gene and make the sequencing to check the accuracy of cloning, so if the gene size is long, then we use forward primer sequencing and reverse primer sequencing. Company gave us the results, sequencing results, so now we need to merge the results of forward and reverse primer sequencing and check that our cloned gene is right or have some mutation or we did not clone the right gene. For this purpose, we use the Bioedit tool to make the forward and reverse sequencing results to make a consensus or conserved sequence that we have already downloaded from any source like cytosome or any other, and then we use to clone it. So we will compare with that we have cloned that gene and our sequencing results are 100% match with that or not. So Bioedit is very helpful for determining such type of results and give the quick results. It is a free tool and you can easily download and use it. So how we use the Bioedit to make our forward and reverse primers consensus? Let's start. So this is, I am working on GMAT2 gene, and this is the sequence that I have downloaded from the cytosome So then I have made the forward and reverse primer to clone this gene, and then after the cloning I have sent that samples to the company and they asked them to do the forward primer cloning and reverse primer cloning. So I have got these results from company. This is the result from forward primer and these are the results from reverse primer sequencing. So we use the Bioedit to make conserve or merge them to make the one sequence. So for this, open the Bioedit, then go to file, open, and then open that forward primer file. So you will get this type of sequence. This is your forward primer sequencing results sequence. Now we need to upload or open the reverse primer sequence. So if remember that if you again go to file and open that file, so it will open in new window like this. So every sequence that you open through that file and open, so it will open in new window. So it is not good. So make them in or open them in one window. So you need to go to the file, then here import and sequence alignment file, then select that, and here it is your reverse primer and forward primer in one window. Now we need to make the consensus or conserved or make the complete sequence or merge these primer and reverse sequences in one sequence by gene editing or removing the mismatches within the base pair. So we need this reverse primer to its reverse complement. So you need to make its reverse complement, this reverse primer sequencing into reverse complement. So select that reverse primer. Here I can show you this one. So reverse primer, select reverse primer and then go to the sequence and select here nucleotide, and then here this is the reverse complement. Click on that reverse complement, so your reverse primer sequence results change into its reverse complement. Reverse complement. So now we need to check the alignment of these both genes and make that consensus. So we need to select these both and go to the sequence, and here is the pairwise alignment. You need to select here pairwise alignment, and then here is the option align to sequence, allow ends to slide. Select this one. So less than two sequences are selected. Oh sorry, we did not select the both. Here select it. Press the cursor on one sequence and click, and then press the control and press on another sequence. So in this way you can select both the sequences, and then sequence pairwise alignment, align two sequences, allow ends to slide. And here is the some parameters of or results of that alignment. So minimize it, and this is your sequence alignment. That here this is it shows the forward primer. Here these are the forward primer alignment, and when we scroll it into the right side, so it gives the some mismatches between these two sequencing results, reverse and forward. So here this one. So this is we have done the alignment. Now we need to make them consensus or conserved, or merge them in one sequence. So for that we need to go to the alignment toolbar, and then here click on create consensus sequence. So here our both forward and reverse primer sequencing results merged into one sequence that we can use in further analysis. But before this we need to make the alignment of this consensus sequence with our that sequence that we have downloaded from Cytosome, to check that it is similar or identical to that sequence, to know that whether we have cloned the right sequence or not. When we get that alignment 100% okay, then we can say that our cloned gene is right, and we can use it in further expression analysis or any other analysis to characterize that gene functionally or experimentally. So for that you can keep your cursor here at the first base pair. I show you here. You can keep the cursor here and then press it. Now go to the edit and select to end. Click on it. So your sequence will be selected. All this sequence is selected from start to end. So you need to copy it. So go to the edit, copy, then open the file, txt file, and here you can make the consensus sequence. So here is consensus are conserved, and paste it, save. Now we need to make its alignment. So go to the Google or any search engine Then write the Genome GP. Genome GP. You can also make this alignment in phylogenetic tree or in Mega or any other. So I usually use this because it is very easy and gives the quick results. Then open this Genome GP, the first link. You can open it. Now click on here, this one at the below. There is a clustalw. So click on it. So first I need to that copy that my sequence that I have downloaded from Phytosome. Copy it, and here you can paste. Then I need to copy my that conserve or consensus sequence from my cloning and its sequencing results. Paste and then execute multiple alignment, but here you can select DNA and execute and multiple alignment. So here are our results. Now this sequence is our sequence that we have downloaded from Phytosome. So this is the primer sequence that company used to clone or make the sequencing of that clone gene, or you can say this is your vector primer, like p-donor or like this. So this is the sequence of our gene. So it is really matched. So we need to remove this primer sequence from our that conserve or consensus sequence. So it is easy. First you need to, from here, this one is our start codon. So from here you can select any these base pairs. Before that ATG and go to the that consensus folder and control F find, and then control V and find next, up. So here this one. So select this all before that ATG and delete it. Now we need to remove that end primer. So this is a TGA or a stop codon. You can see here, this is the TGA or stop codon. So we need to remove from here T. So copy and then find it in our consensus sequence, or we down next. This is, so here is the TGA and remove it or delete it. So now it's our sequence without the primers. So we can check it again for making the alignment. So copy this sequence that we have just made before the after the removing the primer. Sorry, copy it and then here we need to paste our sequence in the consensus region, because we have, and then click on execute multiple alignment. So now our alignment results come. So it will start from that start codon. So it gives that about 100% similarity or identity to that stop codon. So in this way you can use the Bioedit to merge the sequences of forward primer sequencing result and reverse primer sequencing result to check your clone gene that it is 100% right sequence or right clone or have some errors. So then after the confirming, then you can use it into further experiments to make it characterized it experimentally too. So hopefully this video helps you. Thank you.